Welcome to 9.2, Diversity in Animals. What we're going to look at is different evolutionary relationships and their structures of animals and their processes. So some of the processes that we're going to look at are regulation, neuron function, and cardiovascular system, just to name a few. So let's jump right into diversity of animals and start comparing different phyla and evolutionary relationships in terms of their structure and function.
And what I want to talk about is some key characteristics of animals. And something that all animals have in common is that they are heterotrophs. They have to get and consume all the molecules they need for energy and their basic building blocks through consuming other organic molecules. So they need to eat other plants, other animals, other decaying plants or animals, like with their detritivores or detritus. Animals are going to be motile. That means they can move around. Now, some are not going to be mobile throughout their entire life. Some will only be mobile during their larval stage, whereas other animals are going to be mobile during their entire life. Animals also have specialized cells and tissues to complete specific functions or cell specificity. And this leads to more functions, more variety, more adaptations. So specialization allows for more structure relationships. And all animals are going to be multicellular. So what we're going to look at is we're going to look at groups of animals and some key events in animal evolution. The simplest animals are going to be the sponges in phyla Porifera. They have specialized cells, but they do lack specialized tissues. And in their adult state, they are sessile. However, in their larval state, they are free swimming, and that's where they are going to be motile. At one time, they thought that sponges were plants, but upon closer identification study, they were determined to be animals because of their feeding mechanism. They are filter feeders. They do excrete waste. They do have a sexual life cycle. So they do have all the characteristics of animals. They are just the simplest. Next, we have phyla Cnidaria. Here we see that they have radial symmetry. They have a radial body plan. They do have tentacles. They all have some form of tentacles. Your jellyfish and sea anemone and hydras fall into this group. Your Portuguese man of war is a colonial made up of many different cnidarian organisms. They do develop from two embryonic cell layers, and their tentacles that they do have stinging cells, which is what their knit phyla is named for. So the gastrovascular cavity that they have is kind of the first kind of what we would consider an organ system in a way. Specialized sac for digesting food. However, that's about it for cnidarians. Okay, but they are a very interesting species as they capture their prey with these little harpoon-like cells. Some of them can be very toxic to humans, just with the box jellies. Others just give you a very bad sting if you come into contact with them. And some of them are harmless. So some other key events that occurred in the evolution of animals. It begins with the invertebrates. So we have bilateral symmetry, which we start with in the flatworms. This is where your body can be divided into left and right sides. Then we form three different tissue layers. We get embryonic germ layers, ectoderm, mesoderm, and endoderm. And these germ layers are responsible for developing into some key tissues and organs. So for example, Your ectoderm is responsible for developing your outer skin and your nervous system, the nerves. Mesoderm, muscle, skeletal system, vessels of the circulatory system. Endoderm, on the other hand, is going to develop into the lining of the digestive system. Then we have body cavities, and we're going to look at these. The body cavities are known as coeloms, and they are fluid-filled, and they have membrane-type tissues that line both, not just the organs, but also line the cavity itself and suspend those organs and support those internal organs. It's to prevent, it allows for the organs to move with the animal. It allows those organs to be protected from any sort of shock. It's kind of like the fluid is kind of like a shock absorber, if you will. So let's look at what we consider a coelom. Well, once again, here's your flatworms. Everything kind of sort of happens with invertebrates in the worms. With flatworms, we have our three types of tissues. We have bilateral symmetry, but we do not have a true coelom. Notice you have ectoderm right on top of mesoderm, right on top of endoderm with the acoelomate. Okay, the digestive cavity, okay, where their food goes to be digested, is the only cavity that they have. Everything else is surrounding them is a mass of cells. Then with nematodes, our roundworms, we have kind of sort of a coelom in that light pink. We call that a pseudocoelom because it's not completely lined with endoderm or mesoderm. Okay, you have the coelom, and then you have the mesoderm, you have the endoderm, and you have the ectoderm. With the eucoelomate, we have the coelom, but we have the suspension. We have that mesoderm all around and attaching to the digestive tract. So it's kind of suspended, and that is in the annelids, in the segmented words. That's where we first see this true coelom. So it's just going to kind of give that digestive system and those organs a suspension, and so they can move around with the animal.